So I'm very excited to be talking to Gary Phillips, the CEO of Pharmaxis today, about their new drug that I think could be a real game changer for people with arthrofibrosis, both in prevention and treatment of it. For those who don't know, arthrofibrosis might be the most common disabling and painful disease that your doctor has probably never heard about. So to understand how this new drug works, we need to get a little bit technical. Arthrofibrosis means scar tissue inside of a joint, and it makes the joint stiff and painful. And it's called by a whole lot of names, called stiff knee, stiff elbow, adhesive capsulitis, frozen shoulder. They're all basically the same pathology. And it's a little bit like an internal burn scar, but instead of a burn being the instigating factor, it usually begins after an injury or surgery or both. So this scar tissue is made of collagen, and collagen is a kind of protein that the body makes. And it's made by a special type of cell called a myofibroblast. It's a bit of a mouthful. But these cells are only called into existence to heal wounds. And after that, they're supposed to disappear. But in fibrosis, they become a little bit like zombies and they won't go away. And that leads to the pathology that we see in fibrosis. So not only do the myofibroblasts make the collagen, but they also need it to survive, and they use it like ropes to pull and contract the tissues around them. So to get to the myofibroblasts, we can target the collagen. And this is what Gary is here to talk to us about. So Gary, could you please tell us about Pharmaxis and what the drug is called?
Sure. Yeah. So Pharmaxis is a relatively small biotech based in Sydney. We're slightly unusual in that we have an in-house team of drug discovery groups. So we have biologists and chemists working in labs with white coats and safety glasses and walking around with pipettes and working in extraction hoods and things. So you'll find a lot of biotechs around that a lot of this sort of research work is outsourced into universities and whatever. But at Pharmaxis, we've believed it was worthwhile developing a team, and they're made of, it's like United Nations when you walk into our labs. The group is run by two Germans, Wolfgang and Dieter, sort of head of biology and chemistry, and and they've really been responsible for the breakthroughs we've made in looking at enzymes which tackle this fibrosis topic. And I hesitate to say we're sort of global leaders, but I think in this particular very narrow area, this small little company in Sydney is actually a global leader. We've had 3 publications in Nature magazines and Nature publications in the last couple of years with collaborators that we work on globally. And it's really, resulted in this the drug which we're here to talk about today, which is PXS-5505, which is a drug which inhibits lysyl oxidase. And lysyl oxidase is inherently involved with that the myofibroblast that you talked about earlier. So I'm happy to sort of go further and get into the science if you'd like to on 5505.
Sure. Yeah, maybe you can just tell us about what this LOX enzyme does.
Yes. So I think of it in terms of maybe some of your listeners used to have an analogy, I think, at sometimes. And if any of us ended up hurting ourselves with a cut on the skin, for example, then the body often responds after the wound healing process by flooding the area with collagen fibers as a part of the process by which the wound gets closed and a scar forms, which gives it integrity back to the skin. And you can think of those fibers as a bit like for those of you used to mending holes in speedboats or cars with fiberglass, it's kind of the fiberglass mesh that you can buy from a DIY store, and it's floppy and loose and it looks like just strands of wool or cotton. But those strands and that mat of floppy mesh actually becomes stiff when you add a hardener to it. And that, in a sense, is what the lysyl oxidase enzyme is. You can think of it in broad terms as a hardener. So the myofibroblast that you talked about, under certain circumstances, as you said, through an injury, through surgery, it could be a genetic problem that a patient has. It can be a chemical insult, any number of things that can lead to the myofibroblast becoming activated. And when they do, they secrete collagen fibers and lysyl oxidase. Lysyl oxidase is actually a family of enzymes. There's 5 of them altogether, which we name LOX, and then LOX 1, 2, 3, and 4, helpfully. So when the LOX is in the presence of the collagen, then it forms cross links between the collagen. So you can imagine all these fibers now getting little links between them, and as those links form, facilitated by the enzyme, then they form a stiff tissue, and the more cross links there are, the stiffer the tissue gets. And your body has a kind of a feedback loop. So as the tissue becomes stiffer, it tends to cause an increase in the activation of more myofibroblasts, which then cause more collagen and more LOX to be secreted, and therefore more scar tissue. So this whole process goes on and on. Now, in a healthy situation, and if you, again, I go back to your skin maybe and looking at a scar on your skin. If you have a scar, all of us have got scars, but they tend to look the same year after year after year. They don't look like they change, but actually underneath, the tissue is being rejuvenated all the time. So the body eats through the existing fibrosis or scar tissue and chews it up. On one side, so it's degrading it. On the other side, it's still producing myofibroblasts with collagen and LOX as well. So your scar, although it looks the same, it actually quite a high percentage of it is changing every year as some of the scar tissue is eaten up and new scar tissue is laid down in exactly the same format. So you've got this balance. In a situation like arthrofibrosis, that can get out of kilter. So the rate at which the body is chewing through existing scar tissue is slower than the amount of new scar tissue which is being formed. So over time, the amount of scar tissue builds up, and we end up with an excess of scar tissue, which then causes problems with function, and obviously goes on to cause all the other symptoms of pain. And this process is the same whether we were talking about fibrosis in the skin, in a joint, in the kidney, your heart, your liver. Exactly the same thing is happening. It's all about the collagen, myofibroblast, lysyl oxidase. You'll find that everywhere. So what our drug is aiming to do is, by inhibiting the lysyl oxidase enzymes, you can slow down the rate of formation of new fibrosis, and therefore the body's natural tendency to chew through existing fibrosis starts to get the upper hand again. And in the end, it comes back into balance. And therefore, when you use our drug in models of fibrosis, you actually can measure a decrease in the amount of fibrosis that you find. We can count the cross-links in the collagen, and we can show that we reduce the number of cross-links in the collagen. And all of that puts the system back into balance, and therefore you end up with hopefully a healthy level of scar tissue and not an unhealthy one.
So how is this drug administered?
5505 is administered orally. So you take, it's a tablet, or actually a capsule, which you can take twice a day. And we've tested it in healthy volunteer studies, and in that we have some assays which we developed as well, which allows us to measure the amount of lysyl oxidase activity in the blood, and we can show as you increase the dose, the amount of activity of the lysyl oxidase goes down. And if we give a dose of 200 milligrams, which is the same as an ibuprofen tablet, 200 milligrams twice a day, we inhibit the amount of activity of the enzyme by more than 90%. Or throughout 24 hours. So we know we have a very effective drug that inhibits the enzyme in the blood. In those healthy volunteers, we also took skin biopsies. So we looked at punctures of tissue through the skin, just to see if we were getting penetration of the drug into the tissue. And again, we can measure reduction in the enzyme activity in those skin biopsies as well. So it really shows the drug it's a very small drug. It's not what we call a biologic. It's not an antibody. It's a small molecule drug, and its penetration, it goes everywhere in the body. So it's we believe It's a very effective drug at inhibiting lysyl oxidase, an enzyme, and it's also in those studies so far has proven to be really safe as well.
That's fantastic. That's very exciting development. So, what have you trialled it in so far? What conditions?
So we started off. So we looked at fibrosis, as you've just explained, is a phenomenon that comes in, all over our body. So we're sort of sport for choice about where we would look. One of the earliest collaborations we had was with a group in Boston that were very interested in bone marrow fibrosis. Now, in that form, it's kind of a cancer of the bone marrow, where the fibrosis or scar tissue takes over the bone marrow, and the bone marrow in the body is responsible for producing red cells, white cells, and platelets. So if you have a fibrosis in your bone marrow, then you effectively become anemic. Your platelets go low. You're more prone to infections because you've got less red white cells as well, and eventually you develop leukemia. So these patients have about a 5-year life expectancy once they're diagnosed. So that was a really attractive area with a very high unmet need. These patients really didn't have anything to help them other than symptomatically. So the first study we've done is in those patients, and we've had them on. I think we've now completed. I think we've recruited 22 patients. 10 of them have completed more than 6 months of treatment. And what we've seen so far is a remarkably well-tolerated drug. So we're not seeing any serious adverse effects of the drug at all over 6 months. And we're seeing, I think we had 9 of those 10 patients were kind of evaluable in terms of you could measure their bone marrow fibrosis. And 5 out of the 9, we could show a reduction of at least 1 grade in fibrosis in their bone marrow and start to see improvements in red cells and platelets as well. So a very good safety profile and an encouraging sort of early signs of efficacy. So the drug certainly does what it says it does. It does reduce fibrosis. And now it's a question of identifying other areas and other diseases where that anti-fibrotic effect can be used for the benefit of patients.
That's fantastic. So I guess if you're applying it in situations where there's mature, strongly cross-linked collagen, it'll take longer to have an effect as opposed to freshly formed scar tissue. How long do you think it might take to be effective in someone who's had arthrofibrosis for years?
Yeah, so that's a really good question, and I'm not sure we actually know the answer. We did a study recently with Professor Fiona Wood in Perth at UWA, and that we had a similar drug, but one that was applied as a cream. And we looked at patients who had an established scar on their skin, and the drug was applied 3 times a week. So not every day. It was applied once a week, 3 times a day. 3 times a week, once a day for 3 days. And in those patients, they had an average age of their scar, which was 13 years. So these are patients with old scars. This was a placebo-controlled study. So half the patients were taking a cream with our active drug in it, very similar to 5505, and the other half the patients were taking a cream that had no active in it at all. And then at the end of the 3 months, we took skin biopsies of those patients, of the scar tissue of those patients, and then we looked at the amount of collagen in the biopsies taken from those patients after 3 months. And we could show a 30% reduction in collagen in the patients that were on the active drug versus the patients that were on the placebo. So even in a scar which is 13 years old on average, I mean, some of these patients had a scar actually that was, I think the oldest one was, I think, getting on to nearly 50 years. Wow. So it was a really broad range of patients. And this was, I think 42 patients in total. So 21 on active, 21 on placebo. So even in that group, we saw a really big change in the structure of the scar. So I think, when we look at the bone marrow fibrosis one, we are seeing reductions in fibrosis at 6 months. In the skin, we're showing differences in collagen and therefore probably the fibrosis at 3 months. So I think that sort of order, I mean, we're going to follow these patients out to 12 months to see whether there's a continuing improvement. So how long does it take? But your question is a really good one. And it's also, it would appear that different parts of the body, and depending on your age and how old the injury is, their collagen and the scar tissue turns over at a different rate. So you would expect somebody that's older, with a far older injury, maybe the turnover would be slower, and therefore it would take longer to rebalance this fibrotic process. But it really is still something we need to understand and need to trial in patients to find out what actually happens there, and obviously how much of a reduction do you need in order to produce a clinical benefit? I mean, you're not interested. Most of the patients aren't interested if you've reduced my fibrosis by 10%. Is not interesting. Have you got rid of my pain? Is the interesting question, right? So it may be that you don't need to impact the fibrosis that much in a joint per se in order to get a functional benefit for that person, or maybe you need to do a lot. It's a question we still need to understand and research.
We haven't had the opportunity to test these questions before. So.
Yeah.
So yeah, terrific. So do you think this 5505 can actually cause the myofibroblast to go away over time?
Well, I think it's about the myofibroblast activation more than the number. I think they're there all the time. It's really a question of what state they're in, and as they become activated by environmental factors and injuries and surgeries and things, then they become activated, and that's what causes the problem. If we could, maybe there are other ways of turning them off, but as I said, one of the things which stimulates and further activates them is the stiffness of the tissue in which they're embedded. It's a mechanical stress on the cells which causes at least one major factor in their activation. So if we can, by reducing the enzyme activity, we can make the tissue softer, then that's a positive feedback loop, and you'll see less myofibroblasts becoming activated. So I think that's our understanding of the biology at the moment, and that will no doubt be informed by more research in the future. But that's where the state of play at the moment.
Excellent. So people would probably be on their anti-inflammatory medication at the same time.
Yes, I think that fibrosis is the end stage of an injury process. If you have an injury, the first reaction of the body is inflammation. And then if the inflammation is chronic, then it becomes fibrosis. I had a useful analogy. I had a liver specialist who I was once introduced to a room of people to talk about one of our drugs, and he was talking about liver damage to the liver, and he started out with an analogy using the skin, like I did. He said, well, if you stick a pin in the back of your hand, it will bleed, and then the next day you'll probably have a red mark on your skin, and then within 2 days it's gone. Scab's formed, it's fallen off, it's okay. He said, but if you stick a pin in your hand for 7 days in a row for a week, at the end of that week you'll have a very inflamed area which is much bigger. He said, and if you carried on doing that for a month, you will eventually end up with scar tissue forming where you keep on repeatedly stabbing yourself with a pin in the back of the hand if you were so foolish to do so. And so you go through this chronic inflammatory process which eventually ends up in scar tissue. And his point was, that's what you do with your liver when you drink every day. So it was his argument as to why you should give yourself an alcohol-free break during the week, because you remove the stimulus, and therefore you don't get to this point where you get fibrosis and the scar tissue forming in your liver and liver cirrhosis that end stage sufferers have. So I think that's a useful way of thinking about fibrosis and inflammation. The inflammation comes first, the fibrosis follows it. So probably using an anti-inflammatory together with something which is reducing the ongoing fibrosis would be quite a useful combination to get the most rapid resolution of symptoms that these patients are suffering from.
I think that's a really nice analogy, actually, because in knee arthrofibrosis in particular, the Hoffer's fat pad or infrapatellar fat pad, IFP, it swells up and it gets pinched between the bones because it's swollen. So every time someone's standing or walking, it's like that pin being stabbed into the back of the hand, and it's constantly aggravating that inflammation and then the fibrosis. So hopefully we can intervene in that process.
Yeah, it's just a question of rebalancing and trying to find a way. But so even in the other conditions that we're looking at, the bone marrow fibrosis I talked about, we think the drug is best used in combination with other therapies which relieve other symptoms. Removing the fibrosis is not an overnight thing. It isn't like taking a paracetamol or an ibuprofen for a headache. It's really, you've got to be, we think, on treatment for a matter of months rather than weeks in order to turn about a resolution for it.
Yeah. So for someone like myself, for example, who's had it for a very long time, it might even go out to a year. Do you think that we'd need to be on?
We don't really know. We'll see. And I think the nice thing is that the drug, we've had patients on drug for out to a year, and we haven't seen any side effect. It's fine. If the drug works, you take it for a year, it's great. But of course, drugs are, they do impact on our bodies, and they always come with other, sometimes unwanted effects. So you need to watch for that. And I think it's great that our drug has already got a good safety profile. The FDA, the US regulatory authority, were quite happy for us to go into a further study in these patients for a 12-month period. So that's sounds well. I think probably the other thing is to think about is that with this type of injury, depending on the percentage of people who get an arthrofibrosis after surgery, then you might well use this drug in a preventative fashion. So rather than people using it once they have some fibrosis there which is causing them problems, if there's quite a high percentage of people who are going, we know are going to get that as an after effect of the surgery, and if the drug is safe, then there wouldn't be a downside for people to use it to stop that fibrosis emerging in the first place, probably just while the healing process is taking place and there's ongoing inflammation. Once once that surgery is settled and the joint settled down and the inflammation is gone, you could stop treatment. So it may only be a short, you might only be able to treat it maybe for 2 or 3 months after surgery, which would then set you up for a much better long-term outcome for all those surgeries. So I think there's a number of different modalities we could think about how to use the drug in those patients.
Sure. The main treatment to date is surgical cutting of the scar tissue just to release that tension. So this could be used after that potentially to prevent the reformation. Yes,
because once you've got it once, most it's like patients that scar badly. There's plastic surgeons know that this patient is going to scar. Every time I operate them before, they've scarred, and they can do what they want, but they are very likely to scar again. So there's also a population here which we probably are at high risk, and again, worth thinking about how we treat them.
Sure. So that leads us into some questions that I've had from the International Arthrofibrosis Association members. So our first one was, the article said it could possibly treat established arthrofibrosis, but what would it actually do with the scar tissue material?
Yeah, so so if you think of this as a balance, so you've got your body is naturally digesting scar tissue, and as it does so, it breaks down and cuts up those protein strands, which then get broken down and excreted by our bodies. What our drug is doing is in stopping the new scar tissue forming. So you're going to end up, maybe you might end up with more non-cross-linked collagen to start with, because you're stopping the cross-linking process. But again, non-cross-linked collagen is far more easily digested by your body as well. So it's there's not really an issue with where does the old we're not leaving behind a lot of litter behind us that needs to be cleared out. I don't think.
The body's very good at cleaning up generally. So how does the drug target the scar tissue and not the collagen that's normal that should be there?
Yes, a very good question. So one of the things that we looked at very early on was the collagen that you find in connective tissue in our bodies. So, cross-linked collagen forms a basis for, our blood vessels, the aorta, tendons that we have. So it's a fundamental housekeeping enzyme. So you go into this with some trepidation, saying, well, if I stop doing that, then what's going to happen? Am I suddenly going to get, all my joints become loose and my tendons get longer and I get stretchy? And so we did a lot of work on that early on, and it would appear that once we reach adulthood, the collagen that we've laid down in the sort of the connective tissue in the rest of our bodies, which is essential, has an extremely slow turnover rate. We can measure it in decades.
Wow.
So inhibiting lysyl oxidase, which stops the formation of new thing, but doesn't speed up necessarily the digestion of the old tissue, really doesn't have any impact at all on these tissues. And we've done a number of in-depth investigations into this that we've talked through with the regulatory authorities like the FDA, so to get them comfortable. But certainly in patients that are already adults, and we're talking about treatment periods of anywhere up to a year. We've already used it, and we don't see any, as I said, no tolerability issues at all. So yeah, it's a very good question, because it's theoretically it could be an issue, but thus far, based on what we understand about human biology, the nature of the drug, and the experience we've had from patients, is that this is not going to be a problem. I think you'd be cautious about using it in patients at the moment who are still developing and laying down that connective tissue. As I say, once you reach adulthood, that doesn't seem to be a problem.
That's fantastic. Very interesting. So you mentioned the FDA a few times. When do you think that FDA approval might be, realistically happen?
So it depends on which indication.
Yeah.
So we're doing what we call a phase 2A study in myelofibrosis, which is the bone marrow cancer at the moment. That's likely to take us until probably, I think our next discussion with the FDA is likely to be at the end of 24. After that, we'll do the pivotal study, which will lead to an approval, let's say, within 3 years of that. So in myelofibrosis, we're probably about 4 years away from an approval, I would think, in that sort of order of time frame. With the arthrofibrosis, it then depends on, we've got to get it to show that it works. I think the difference between these diseases is that arthrofibrosis, from what you say, is a very, very common condition. Therefore, and there don't appear to be an awful lot of treatments available at the moment to help those patients, other than painkillers and anti-inflammatories, or repeat surgeries. So it's likely that if we did engage in a clinical trial program, that the recruitment would be fast. This is one of the main problems in getting drugs to market, is the speed at which you can run a clinical trial to test whether your drug is both safe and effective. And the work we're doing now is helpful, because it's demonstrating safety. And some of the patients that have myelofibrosis probably also have arthrofibrosis somewhere. We should be asking them maybe, but yeah, it would be interesting. But I think the development process could be quite quick, because there are so many patients available.
Yes, there's certainly been a lot of interest in this drug, I can tell you that. So how are we going to measure effectiveness, or how do you measure effectiveness of the drug?
Well, there are two main ways. I mean, the first one is we always like to show that the drug is getting to the site of the fibrosis and changing the amount of fibrosis. So, in things like scarring on the skin and in bone marrow fibrosis, we're able to take biopsies with a needle to take tissue out, and then we can, by looking at that tissue through a microscope, a pathologist can assess the grade of fibrosis that's there. So that's that's one way. We can also look at the amount of collagen or the amount of cross-linked collagen in those tissue samples as well. But as I said at the outset, most patients are not really interested in what percentage fibrosis they have. Are they in any pain? So I think in myelofibrosis, we're measuring their patients' symptom scores, which is a bank of questions around all the symptoms they get from their disease. And we're also measuring their blood count. So we're looking at the sheer volume of red cells, white cells. So what are the clinical benefits of the drug? In arthrofibrosis, I suspect we'll be looking at more quality of life questionnaires, which will assess how people feel about their mobility, pain levels every day, their level of activity that they can engage in. So you're looking at making a significant change to the way people can live their daily lives, I think would be the primary endpoint, which a regulator would be probably most interested in. We could get more technical and look at I guess in certain areas you might be able to look at mobility of a certain joint. But I suspect that everybody's arthrofibrosis is slightly different. The impact on their joints is slightly different. So one patient with a frozen shoulder, I suffered with a frozen shoulder for many years, and I know the symptoms from that would change almost on a daily basis, depending on how I slept the night before, what I was doing, how much activity I had. So trying to say, okay, we recruit 100 patients and they could extend their right arm 4 degrees higher than they could before, because is probably asking for trouble. So I think it'll be dumbed down to the point of asking patients how they feel is probably the way we'll go.
I think in these it's probably a little bit clearer in terms of range of motion, because the knee joint flexes in many fewer ways compared to a shoulder. So range of motion's a fairly standard approach for a knee, and they have the goniometer to measure that, and they're quite experienced at doing that.
Okay.
And you get to what they call a hard stop, where it just won't go any further regardless. So if we can change that, it will have a big impact on daily living, as you said, ability to drive a car or walk up steps, or even just to get into and out of a chair. So very exciting, I think. So another question: how accessible would this be for people in Australia?
Yeah, that's a sort of an unknown question, isn't it? An unknown answer. Anyway, I think I've worked with the Australian government on drug approvals and getting on the PBS for a lot of my career, and I think the chances of making it available in Australia are very high if we can show improvements in quality of life. I think that the PBAC, which is the group which makes recommendations for PBS listing, look at quality of life as a measure is a very clear motivator for them, and also in the absence of anything else working. So I think one of the challenges they have is to say, okay, well, if you bring in a new drug which can help patients with this, what does it replace, and how much are we paying for that? And therefore, how much more is this one worth? So there's a series of questions to go through, but I think it depends a lot on the results that we get. But I'd be certainly very confident that the Australian patients would be able to access it, as long as we can demonstrate it in clinical trials that it works.
Very good. And the last question: is there a percentage of people who respond and who don't prove an efficacy? Again, maybe the one we did with Professor Wood on skin scarring is the best measure of that, in that we had kind of these 42 patients, and half on placebo and half on active, and the average improvement or reduction in collagen in the wound was 30%. And I'd have to look more at the data, but I think the majority of patients are responders. I think you probably are going to see a range, and there are patients, because I think by its very nature, fibrosis is a natural process, and there are patients whose metabolism, for whatever reason, is chewing through the fibrosis that bit quicker, so we're going to be quick. I think everybody probably ends up at the same spot. The speed at which you get there might be
might change. But the cross-linking of collagen is the final stage in fibrosis. There are many stages above it, but the last one that forms the fibrosis is the cross-linking. And we're dealing with an enzyme, a single enzyme. There are no backup systems that the body has for doing that. It's lysyl oxidase or nothing. And we know that we inhibit in all patients that we've tested, we always fully knock out lysyl oxidase. So I'm pretty confident that we would have a very high responder rate if reducing the fibrosis leads to an improvement in benefits for the patients anyway.
It's very promising. I think there's huge amount of potential here to really change people's lives, give them their lives back.
Yeah, no, I'm excited listening to your excitement listening.
And not only for arthrofibrosis, obviously, for a huge range of conditions. It's incredible. Yeah. And I think the work that Pharmaxis is doing is fabulous. Really appreciate what you're doing.
Good, thank you for your time.
Yeah, well, thank you for guiding us through this rather complex topic. It's terrific.
Okay. Thanks very much. Thanks, Kayley.
I'll see you later. Bye.
Yeah, bye now.